Welcome to National 5, Unit 1, KA2 Transport Across Cell Membranes lesson. This is Lesson 2, Part B. Please make sure you've watched Lesson 1, Part A before moving on to this video. Within this lesson, we'll be moving on from looking at diffusion to osmosis, which is the second form of passive transport that you need to know about for National 5. Remember, passive transport is the category of transport of molecules across the cell membrane which doesn't require energy to take place. So in today's lesson, we're going to look at osmosis. We're going to investigate the movement of water through a cell membrane, and we're going to study how a cell is affected by being immersed in high and low water solutions. Your success criteria is you should, by the end of the lesson, be able to state the definition of osmosis and identify plant and animal cell that has been submerged in high or low water solutions. Firstly, we're going to start by looking at the similarities between osmosis and diffusion. There is actually quite a lot of them. There's very few differences though. So both of them are passive processes. Both involve the movement of molecules. Within both forms of transport, molecules move from higher to lower concentrations, and within both, the molecules move down a concentration gradient, as we looked at in this diagram before. The important thing is that the only difference between diffusion and osmosis is that osmosis is just about the movement of water molecules. Diffusion refers to the passive movement of all other molecules such as glucose, amino acids, oxygen and carbon dioxide, but osmosis is a special case of diffusion and only involves water molecules. Therefore, the definition for osmosis is almost exactly the same as the definition for diffusion, as most of the characteristics are still the exact same. So this time, though we put the word water into the definition 4 times, that's the only real difference here, and that's a really good way to remember it. So think of the exact same thing word for word as your diffusion definition, and then make sure you add in water within that definition 4 times. So osmosis is the movement of water molecules from a higher water concentration to a lower water concentration down a water concentration gradient. And then at the end, we also add in and remember that this is across a selectively permeable membrane. So remember, selectively permeable means the membrane only lets certain small molecules through, like water. So typical exam questions around osmosis and tests or exams can be based on the concentrations of substances. So substances can be dissolved in water, and that changes the overall water concentration. This can alter the concentration gradient as well. So in questions, you will see substances such as sugar, sucrose, salt, starch or glucose being dissolved in water, and then from there you need to then work out the concentrations of water. So we're going to look at the first example on the left hand side here. If in the question regarding osmosis they said the solution was 10% sucrose solution, this doesn't help us to consider osmosis, as osmosis is the movement of water molecules, not sucrose. So if we're told the solution is 10% sucrose, we need to work out what percent water it is. Now remember, we can only get up to 100%, so if 10% of the solution is sucrose, then that means that 10% is sucrose and 90% is water. So just a reminder, can't be over 100. So you basically take away whatever they give you for sugar or glucose or sucrose, take away from 100 and the rest would be water. So if we look at the second example on the right hand side, we're being told that this is a 1% sucrose solution. So if 1% out of the 100% is sucrose, we can assume the other 99% is going to be water. Now that's really important when we come to then answer some questions later. This is usually the first step. So now let's use some of this knowledge to answer some questions. So here we have the two solutions from the previous slide, but now they've been separated by a selectively permeable membrane, just like a cell membrane in the middle. So we could imagine, if we wanted to, that one side of this membrane was inside the cell and the other is outside the cell. And a lot of the times in questions they do that, but we don't need to know that for this particular question. So question 1 at the top asks us to predict the direction that sucrose molecules would move by diffusion through the membrane. So they're asking, would it go from left to right, or would the molecules move from right to left? Okay. Now the first thing we need to do in any exam or test question is you have to read the question really carefully, especially if we're looking at diffusion or osmosis, as we don't want to get these mixed up. So this question is asking us about diffusion, says it very nicely in the question, but if they hadn't said that, we would know it was about diffusion because it's mentioned sucrose molecules moving, so it must be diffusion. So that's the only thing we need to focus on in these diagrams is the sucrose, not the water, because we're not talking about osmosis. So now I've shown you at the bottom how I would go about working this out. Now this is quite a longhand way of writing this all out. I would do this in a little bit of a shorter way, but I'll show you again how to set that out later. So from the previous slide's information, we know the left-hand side of this diagram is 10% sucrose, and we know that the right-hand side is 1% sucrose. This means that there is a higher concentration of sucrose on the left-hand side. Okay, so left-hand side has high sucrose, right-hand side has low concentration of sucrose. Okay, so my first tip here is if you were given this in the exam, I would be drawing on this. So I would write HC as high concentration on this side and LC as being the low concentration on this side. I really encourage you to do this for these types of questions. I still do it myself to try and help you. So we know from our definition of diffusion, we now know where our high and low concentrations are, but we know from our definition that molecules like sucrose always move from high to low. So what I would then do is draw an arrow from where I've written HC to the side I've written LC, and in this case, that means that the molecules are going to move towards the right. So it means that they're going to move from left to right by diffusion, and that would be your answer. So now we're going to look at a second question. So the second question here is a little bit different. So again, reading the question carefully, it's asking us to predict the direction that water molecules would move by osmosis through the membrane. Again, they don't have to say by osmosis. You would know if it's the movement of water molecules that it would have to be osmosis that we're talking about. So this time we're focusing on water and not sucrose. So this time what we need to do is we need to use the percentages that we'd worked out in the previous slide. Now, if you were starting this question from scratch, you would need to work that out at the beginning. So if we do that, a reminder, we worked out that the left-hand side, if it was 10% sucrose, it'd be 90% water. And on the right-hand side, if it was 1% sucrose, it would be 99% water. So that's the first stage we need to do is work out the percentage of water. Okay. Now we know that if 90% and 99%, 99% is higher. So there's a higher water concentration on the right-hand side of this membrane and a lower water concentration on the right. So on this diagram, if I was writing an exam, I would usually write HWC on this side, LWC on the other side for lower water concentration. And then again, I know that osmosis moves from high to low, so I would draw an arrow from high to low. And in this case, that would mean the water molecules would be moving from right to left, which would be our answer. So this is just one more example on the next slide. And this is just to show you again how we would do this. So here we have a selectively permeable membrane separating the outside of a cell from the inside of a cell. And this is asking what happens to the water molecules. So again, we know we're thinking about osmosis. Now in this diagram, water is represented by the blue circles, and we can easily see without any percentages being given to us, sometimes it's just a diagram instead of percentages, that the right-hand side, so outside the cell, it has a higher water concentration. So here I've written HWC. The left-hand side has a lower water concentration because there's less water molecules, so LWC I've written on the other side. So once again, we know osmosis, water moves from high to low, so we would draw our arrow from high to low, which would mean water would move into the cell. Now, instead of saying from right to left, we can use inside outside because they've told us where that is in this example. So we would say water would move into the cell by osmosis. So my top tip for these is always draw on the diagrams to help you, whether this is on a piece of paper at the moment if you're learning from online, or whether this is on the actual exam paper or test paper. Use LWC and HWC for high water concentration and low water concentration, or just use HCLC, so high concentration, low concentration, and draw an arrow from high to low to help you visualise. This will come in really handy later on when we get onto some more difficult questions. So now what we're going to do is we're going to go through 6 examples that I want you to try. Now this works best if you draw these on a piece of paper and practice labelling them as HWC, LWC and with the arrows. Here we're going to focus on water molecules moving through osmosis only and not diffusion. So we're not looking at diffusion, just osmosis. We're going to go through these examples and I'll explain them one by one. You can pause and try them if you want to beforehand, but there are some tricky ones in here that I haven't explained how to do just yet. So if you'd rather just watch them, that's absolutely fine. So in number 1, we start with quite an easy one. I've already shown you how I worked it out here. So we have 70% water on the left-hand side of our semi-permeable membrane, or selectively permeable membrane, and 60% water on the right. Out of 70% and 60%, 70% is higher, so this is our high water concentration, and on the right is our low water concentration. Once we have that, we just draw an arrow from high to low, so we know in this case the water would move from left to right. In number 2, this is a tricky one, as it doesn't actually tell us the water concentration, it tells us the sucrose solution concentration. It also uses something a little bit different that I haven't shown you just yet. So it uses the capital M after the 0.5 and 0.3, and this means molar, which is a measure of concentration. This is from chemistry, you don't need to know that, but for some examples that you'll see, it's good to know that this means kind of like percent, but it's molar, okay? So if there's a higher concentration of sucrose in the solution, so 0.5 molar of sucrose compared to 0.3 molar, there is less room for water. So that means that the 0.5 molar sucrose solution would have more sucrose and less water than the 0.3 molar sucrose solution. So what this means is there's a higher water concentration in this one, lower water concentration in this one, and again, water will always move from high to low, so we're going to go right this time. Moving on to number 3, basically there's 80% water in both, and we know that although water can keep moving across the membrane, there'll be no differences in the concentration gradient here, so there's going to be no overall change. Water is going to move back and forward, but there's not going to be a change. Moving on to number 4, we have 0.2 molar starch and 0.1 molar starch. Remember, it doesn't matter if it's starch, sucrose, glucose. If we're talking about water, it doesn't really matter which one's which. So we have 0.2 and 0.1 molar. Again, not water, so we have to actually think about what the water concentration is going to be. 0.2 molar starch, more starch here, less water, less starch, more water. So this side would have a higher water concentration. The left-hand side would have a lower water concentration, and so we'd move from high to low and we'd go left. For this one, for number 5, this is the same even though the types of substances, so starch and sucrose, are different. It doesn't matter. 0.1 for both of them, so they're still the same volume of space for water, which means that both of them will be identical, have identical concentrations of water, so water will move back and forward just like it did up here in the same way. Finally, we have the cell and pure water. Now, really important to introduce here that cells aren't just water on the inside. They are anywhere between about 70 and 99% water. Therefore, pure water, being 100% water, pure water, will have the higher water concentration and the cell will have the lower water concentration. And so the water will move from high to low towards the left. So now what we're going to do is we're going to put all of these information that we've just done into practice by looking at another quick experiment. So this is osmosis in a model cell. Now here we have a bit of background and we have a diagram at the bottom to help. So the diagram shows a model cell that was set up to investigate passive transport through a selectively permeable membrane. So this is just using viscing tubing like we talked about last time. The model cell was left for 20 minutes in the solution and was weighed before and after. So what you can see is outside the selectively permeable membrane, there is water, and on the inside, there is strong salt solution. The 2 questions that you'll commonly get asked for this type of experiment would be to predict what would happen to the mass of the model cell at the end of the experiment. So what would it weigh, because they've weighed it before, they've weighed it after, and explain your answer. So these are really, really common types of questions for this experiment, or for ones that are similar as well. So here we're looking at osmosis, so the movement of water. So the first question is asking us to predict. Now my first tip here is predict questions are common in the exam and they will ask you to predict, they only want a short answer. They literally want you for number 1 to just say increase, decrease or stay the same. In this case, there's a higher water concentration in the pure water outside the model cell. Strong salt solution will have quite a lot of salt, so less water, so low water concentration inside. So water is going to move from high to low into the model cell. And what this means is that the weight of this model cell will increase. So increase is our first answer. Okay. Now the second question asks you to explain this, and this question is really common and usually worth 2 marks in the exam. It's really important that you follow the same structure each time to make sure that you get those marks. Now the first mark is normally given for saying how the water moves if it hasn't said that in the question already. So you would say the water moves by osmosis. So this is showing that you know that it's osmosis and not diffusion. Then you have to point out and explain why you know that this would increase. So we know it would increase because we know where the high water concentration is, where the low water concentration is, and therefore we know the direction it's going to move. Okay? So what we would say here is the water moves by osmosis from a high water concentration outside the cell to a lower water concentration inside the cell. Therefore, the weight increases. So you get 1 mark usually for this bit and 1 mark for saying where the high and low are and the direction the water's going. So you need to mention the fact the water's moving into the cell from the high water concentration outside to the low water concentration inside. So that's really, really important. Now, to help with this, here's a kind of structure you can follow to answer this type of question in the exam. So it's always the same structure, but you change some of the words to fit the experiment. So sometimes the water concentration will be higher on the inside instead of the outside. Or maybe they aren't using a model cell in the experiment, they might be using something like viscine tubing, potato cylinders, something a little bit different. So the most important thing here is to read the question carefully and use the wording of the question within your answer. You can move some of this about. You don't have to say water moves by osmosis at the start, it can be at the end, but just make sure you mention osmosis, where the high water concentration is, where the low is, and the direction that it moves in as well. Okay, so now what we're going to do is we're going to move on to the next part about osmosis, but before that we'll try some quick questions. So pause the video to try them and play when you're ready to hear the answers. So the first question is, what is the main difference between diffusion and osmosis? So diffusion is the passive movement of molecules of things like glucose, amino acids, oxygen and carbon dioxide, whereas osmosis is the passive movement of water only. Remember, they're asking about a difference, so you should be saying something like whereas or but within your sentence and comparing the two. Give the definition of osmosis. You need the full thing word for word. So movement of water molecules from a higher water concentration to a lower water concentration down a water concentration gradient and across a selectively permeable membrane. For number 3, if solution A and B are separated by a selectively permeable membrane and solution A has a sugar concentration of 1% and solution B has a sugar concentration of 50%, in which direction will the water move? So this one is just asking which direction. So is it going to go from A to B or B to A? And then number 4 is asking you to explain your answer. So first thing we need to remember is this is water and not sugar that we're talking about. So solution A, they've told us, is 1% sugar, so that means it's 99% water. Solution B is 50% sugar, so it's 50% water. So solution A, 99%, solution B, 50%, so water's higher in A than it is in B, so it's going to move from A to B. So A to B is the answer for 3. To explain your answer for number 4, we need to use that same structure we talked about before. So water moves by osmosis from a higher concentration within solution A to a lower concentration in solution B. So I've pointed out osmosis, I've pointed out where my high concentration is and where my low concentration is, and I've also mentioned the direction that the water is moving in as well. So now we know what osmosis is and how to do basic problem solving about the movement of water across selectively permeable membranes. We're now going to learn about the effects of osmosis on the different types of cell we spoke about in KA1, so cell structure. So we're going to learn about the effects of osmosis on different types of cell, and I want you to be able to identify whether plant or animal cells have been immersed in high or low water concentrations and use the correct terms to describe those cells. So cells can be anywhere between about 70% and 99% water on the inside and are surrounded by a selectively permeable membrane, cell membrane, so osmosis is happening all the time between the cell and the environment that surrounds it. Now here's a quick problem, just to check we've got this before we move on. So a cell with a water concentration of 80% is placed in a solution of 25% sucrose. Then it asks us what will happen through osmosis. So again, we can annotate the cell, we can draw on it. So inside the cell is going to be 80% water, as it says that in the question. However, be careful not to trip up here. It says it's placed in a solution of 25% sucrose. We're not wanting to know about sucrose, we want to know about water. So this outside the cell would be 75% water. So 80%, 75%. Again, I would then annotate my diagram if I was doing this on paper, and I would put HWC on the inside of the cell, LWC on the outside, and draw my arrow from high to low going out of the cell, which means that water will move out of the cell. Okay? Like that. So in a test or exam, you can be asked about the effects of osmosis specifically on animal cells or plant cells, and it's really important to know the difference between those. So animal cells If there's a question about skin cells or red blood cells, then they're talking about animal cells. Any cell that would be in your body or a normal animal's body would be animal cells. Plant cells would be the question talking about things like onion cells or potato cells. So keep in mind that in a question about osmosis, you need to work out what type of cell they're talking about. So whether it's skin cells, it's animal cells, onion cells, plant cells. Really important to know the differences in how to describe the effects of osmosis on these cells. And that's what we're going to move on to now. This is the most common type of exam question on this topic.
Okay, so what we're going to do is have a look at what happens when you place animal cells or tissues in different concentrations of water. So we're going to look at water movement, the effect of osmosis on those cells. And the first thing we're going to do is just draw and remind yourself of what an animal cell looks like. So remember, an animal cell, basic one, has a cell membrane, cytoplasm, and then a nucleus. We're not going to draw the ribosomes and mitochondria and stuff here because we don't really need to look at those for osmosis. Now we're going to imagine that we take our animal cell, which could be our red blood cell, could be a skin cell, and we're going to place it first of all into a pure water solution. So when you see pure water, that means 100% water. So we're going to take our animal cell and place it in 100% water. Now remember, the inside of the animal cell is roughly between 70 and 99% water. For this case, we're just going to say it's 98%. So now we're looking at our cell. Inside of our cell is 98% water, outside in the pure water is 100%, so we're going to label our water concentrations. So our high water concentration is outside, our low water concentration is inside, and we know that osmosis moves from high to low, so it goes into the cell. So what will happen in this case is you need to think of the animal cell as being like a balloon. The water is going to move from the high water concentration outside into the low water concentration inside by osmosis, and this is going to keep happening until this swells up and up and up, and eventually the animal cell will burst. The way I draw it bursting is I just draw the outside of where the cell membrane used to be with the nucleus still intact. So we say it bursts. Okay. Now if we take the same type of animal cell, and this time instead of placing it in pure water, we place it in, say, a 2% salt or sugar solution. Now this time that would be 98% water, which would be the same as the inside of my animal cell. Now what this means is there's the same concentration on the inside and outside, so water will move back and forward across the membrane, but nothing will happen, and the cell will stay the exact same. Okay, finally, we're going to take our animal cell, and instead of placing it in pure water or 2% salt, we're now going to imagine that we've placed it inside a strong salt solution or sugar solution. Now sometimes they'll tell you what the concentration is, sometimes they won't. For this sake, let's imagine that it's 20% salt. So if it's 20% salt, that means it would be 80% water, which is what we're going to focus on, and inside the cell it would be 98% water, as we discussed before. So in this case, if I label my high and my low water concentration, it's a high water concentration inside, low water concentration outside, and we know that we draw our arrow from high to low, so water is going to leave the animal cell in this case. And what that means is the animal cell, like a balloon, if you take water out of it, is just going to deflate, so it's going to shrink. And the way I like to draw that is I just draw it kind of shriveling in on itself with its nucleus still there. So the main thing to remember here is in osmosis in animal cells, if you place an animal cell into pure water, then it will burst. If you place it in something that's the same solution concentration in the inside of the cell, it'll stay the same. And if you place an animal cell in a strong salt or sugar solution that has a higher concentration of salt or sugar than the inside of the cell, water will move out and it will shrink. So you need to remember to use the words shrink and burst for animal cells. So now we're going to move on and we're going to look at osmosis in plant cells and tissues instead. And this is quite different than the one for animal cells. So once again, we're going to start just by drawing our plant cell. So remember, it has a cell wall on the outside, cell membrane on the inside, vacuole, and we've got our nucleus. Now also there's the chloroplasts and stuff as well, but we're not going to draw those again because these are the structures that are the most important for osmosis. Okay, so this time we're going to do the same as we did with the animal cell, but we're going to take the plant cell and we're going to place it again in a pure water solution. So remember, pure water, meaning 100%. And then inside our cell, which I'm just going to draw at the moment, is just being a square. Remember, that's about 98%. Now again, if we label this for high and low, high water concentration outside, low water concentration inside, so water's going to move in. Now the big difference here is that a plant cell has a cell wall around the outside. And because of this, you can imagine the cell wall is keeping the structure of the plant cell. So the cell wall is going to stay the exact same. It's not going to burst in this case like it would in an animal cell. What happens instead is water does move into the cell, and because of that, the cell membrane pushes up really tightly against the cell wall. And also the vacuole in the middle becomes more filled with water, so it also increases in size. Now you can see the difference from this compared to our original cell. We can't say it's burst because it hasn't, so what we use to describe this look is turgid. So when a plant cell is placed in pure water, we say the effect is it becomes turgid. Now it's just the same between osmosis in animal cells and plant cells in terms of what happens if you put it in a solution that's the same on the inside and outside. So again, if this was say 2% salt or sugar on the outside, about 98% water, and that was the same on the inside, again, water would just move back and forward like usual. And so the plant cell would just stay the exact same as it was at the start. Now finally, if we take our plant cell and we put it in a strong salt solution, so strong salt or sugar solution, meaning it's very concentrated. Okay, so this time I'm going to use a different concentration maybe than I used for the animal cells. So let's say this time it's only 10%. So 10% salt in this solution. And our plant cell is here. So our plant cell is 98% water. 10% salt would mean that there would be 90% water on the outside. So this means that our high water concentration is inside, low water concentration outside, so water moves out. Now again, we need to keep in mind that this cell wall structure is different that the plant cell has compared to the animal. So again, cell wall is going to keep its structure, but this time water is moving out. So we need to be able to see that in the cell. So what actually happens is the cell membrane, because water is moving out, pulls away from the cell wall. So it pulls in on itself and pulls away from the cell wall. It's not this nice box shape like it was before. And our vacuole, because it's losing water, also gets really, really small, like this. And the name we say for this, we don't say it shrinks because the cell wall still is keeping the structure on the outside. Instead, we say this is plasmalized. So it's really important to remember that with plant cells and osmosis, if they get filled with water, so pure water on the outside moves in, they become turgid, where the cell membrane pushes up against the cell wall, vacuole gets larger. If it's in the same type of solution, concentration solution, then it stays the same. And if it's in a strong salt solution, then the water is going to move out, the cell membrane pulls away from the cell wall, the vacuole becomes smaller, and we call this plasmalized. You must remember the differences between both of these. We need to remember that it's only animal cells that burst or shrink, and it's only plant cells that become plasmid or plasmalized or turgid. So remember the differences between these and don't mix them up or you'll lose your marks.
Okay, so in osmosis in animal tissues or cells, if the animal cell is placed in pure water, so 100% water, water will move from a high concentration outside the cell to a low concentration inside the cell, and then eventually the cell will basically blow up and up like a balloon and eventually burst. So if we get asked the effect of an animal cell of being placed in pure water, the effect is it will burst. If they say effects in a question, again, they just want the word, what's going to happen to it. If we place the animal cell in a solution which has roughly the same water concentration than the inside of the cell, so here we're assuming that the inside of the cell is 1% as well, 99% water on the inside and outside. So in this case, there would be no change because there's no concentration difference, so no movement of water, there's no change to the cell, it just looks the exact same. Finally, if we place the animal cell in a very strong salt solution or sugar solution or sucrose solution, water will move out of the cell from a high water concentration inside the cell to a low water concentration outside the cell, and therefore the cell will shrink. So the effect of this on the animal cell is it will shrink. So the words you should be using to describe osmosis in animal tissue, the effects, would be burst or shrinks. Now for plant cells, although what happens in terms of water movement by osmosis is the same, so the direction the water would go, the overall appearance of the cell, and therefore the term we use to describe the effect, is very different from an animal cell, and it's really key to remember these differences. So for plant cells, if a plant cell is placed in pure water, so 100% water, water will move from a high concentration outside the cell to a low concentration inside the cell, but because the plant cell has a cell wall, it will not burst like an animal cell. Instead, the cell membrane pushes up against the cell wall and the vacuole becomes bigger, and this state is called turgid. So if you get asked the effect of osmosis in plant cells, tissues, of putting it in pure water, the cell would become turgid. If we place the plant cell in a solution again that is roughly the same concentration inside and outside, again it's not going to change shape, it's going to look the exact same. Finally, if we place the plant cell in a very strong salt or sugar or sucrose solution, water will move out of the cell from a high water concentration inside to a low water concentration outside the cell, and the cell will become plasmalized. In this case, the cell membrane pulls away from the cell wall, which you can see in the diagram here, and the vacuole shrinks, but the overall size of the cell is kept the same by the cell wall. So we don't say it shrinks, we say the effect on the plant cell is that it's plasmalized. So you need to remember, turgid and plasmalized can only be used to describe plant cells. They cannot be used to describe animal cells in osmosis. That's just burst or shrinks. Now to show you what this looks like under the microscope in real life, here's a picture of red onion cells, so plant cells. These are turgid, so we have the cell wall pushed up against the cell membrane pushed up against the cell wall. You can't really see the vacuoles in these though, but you'll see the big difference when we go to what's happened when they become plasmalized. So these are the same cell types, but this time you can see that basically the colour is kind of pulled into the middle, and that's because the cell membrane's pulled away from the cell wall because water's moved out, and you can still see the cell walls on the outside keeping their shape. So now we've reached the end of the osmosis theory. Here's another quick quiz. So as usual, pause the video and attempt the questions, and then play when you're ready to go over the answers. So what happens to a plant cell when it's placed in a pure water solution? It becomes turgid. So do not accept bursts, it becomes turgid. What I should say is for all of these, you're free to draw them out. So draw the plant cell, draw the fact that it's 100% water on the outside, label your high and low water concentrations and the direction it goes in, and then think about what will happen to it. Number 2, why is it different from what would happen to an animal cell? Well, an animal cell would burst because it doesn't have a cell wall. So the cell wall is the difference here. What word can be used to describe a plant cell that's been placed in a strong salt solution? Plasmalized. What word can be used to describe an animal cell that has been placed in a strong salt solution? Just say it shrinks. And what word can be used to describe an animal cell that's placed in a pure water solution? It will burst. If you have any questions about the above, or you didn't get them correct, please rewind the video and go back and watch the portion on effects of osmosis on the different types of cell. You need to be able to answer these questions quite quickly and easily. If you're ready, you can now move on to part C, so video part C, where we're going to look at active transport and also consolidate our learning of all of KA2.